Okay, so I'm not going to get into too many details about Emiquon, as the previous presenters today have done an excellent job of giving us all the details we need. I am just going to mention that Emiquon is a restored backwater of the Illinois River along the LaGrange Reach, and restoration began in 2007. The Illinois River Biological Station has been monitoring fish populations on Emiquon since 2007. So during the first 2 years of restoration, the Nature Conservancy, along with the Department of Natural Resources, stocked an initial 36 species. The photos below are some of those species that we regularly run into while sampling, including starhead top minnows, which are state threatened. So Emiquon is overall a very good fish habitat. And since I am focusing on some of the different sport fish species, this graph is largemouth bass CPUE, or catch per unit effort. And Emiquon is the green line, while pool 13 on the Mississippi River is in yellow, which pool 13 is also known as a good bass fishery. The other lines include pool 26 on the Mississippi River and the LaGrange Reach. So you can see that Emiquon is up there in terms of largemouth catch rates. So again, as we've learned today, the Nature Conservancy does have a water control structure, which allows them to either pump water in and out of Emiquon, or in sometimes in. It's also the only real way that Emiquon is connected to the Illinois River. So then we have to ask the question, how does this affect sport fish? So with water level changes, you have varying effects on vegetation abundance, fish habitat, and prey abundance for sport fish. And so those are going to be some of the big factors that I will be talking about for my project. For the objectives of my study, I'll be looking at otoliths from largemouth bass, bluegill, and black crappie, as well as catch data from IRBS's standard monitoring program to determine what environmental or management factors are affecting biochronology, or annual growth, year class strength, and mortality and individual growth. And if I haven't said it enough, my study species are largemouth bass, black crappie, and bluegill, pictured here in case anybody is not super familiar with fish. We chose these species because they're 3 of the most popular sport fish in Emiquon, as well as in Illinois. There is also a previous study that was done by IRBS staff members where they collected otoliths from these 3 species, and we are able to utilize them for my project. So I'll be able to get a good look at the life at populations at Emiquon. So sampling is, I'm sure everyone can guess, it was pretty difficult in 2020. The initial plan was for us to sample from April 1st until May 31st. We chose those dates because that previous project I had mentioned had sampled in April and May, not to mention that springtime is when the fish are starting to move a lot more, they're coming out to spawn, and our catch rate is usually a lot higher in April and May. Due to the COVID-19 pandemic, that didn't happen. I was able to sample that last week of May into about June 13th, and I was able to collect a little over 350 samples, so it's still a very successful effort. So some field methods that we used included boat electrofishing, fike nets, mini fike nets, and tandem fike net sets. These were used because, based on the literature, they're the best gears to use when targeting my study species. They're also the gears used in the standard monitoring program, as well as in that previous project I had talked about. Again, just to quickly go over a couple of these gear types in case anybody's not as familiar. In this top left hand, you have a mini fyke net set. In the bottom right central, you have a fyke net set. Those are set to the shore and ran perpendicular out and then left to sit for 24 hours. And then we also use boat electrofishing for 15-minute runs. And you can kind of see how that process works for the other three pictures. So as I mentioned, we're utilizing previously collected otoliths. We were able to obtain about 14 from a 2010 collection. These were only largemouth bass though. In 2015 and 16, they collected all three of my study species. So overall, I have just over 700 samples for those 4 years to process. Otoliths are hard structures located near or in the brain cavity. And when you section them, you can read them like you would tree rings, which I will show pictures of in just a minute. So these otoliths are embedded in an epoxy mixture, and then we use a Buehler low-speed saw. And I chose to use a double blade setup. So that setup will be, if you look at this biggest picture in the center, it's a blade, saw blade, a plastic spacer, and then another blade. This way you can get a blade on either side of the nucleus of the otolith and get the best possible section for aging and measuring annuli. I was fortunate enough to be able to work with some of the technicians at IRBS, and they really helped me out a lot. So this top picture shows what it looks like after the otolith has been cut. From there, the section can be polished or cleaned, put under a dissecting scope, and then using like a V4 software, we picture it, age it, and then use the measuring tool to measure the distance between each annuli. This next slide is just some pictures of different otolith sections to kind of give everybody an idea of what I am typically looking at. The top left is bluegill, top right is Black Crappie, and then the large bottom picture is largemouth bass. All three of these were captured in either 2015 or 2016. So as of right now, all previously collected otoliths and 2020 otoliths have been processed. So they've been sectioned, measured, photographed, everything. I'm looking forward to starting 2021 otoliths. I also just want to point out to everybody that this picture is a largemouth bass otolith. It was captured in 2020 and it's aged out to being 13 years old. So it would have been one of those initial fish. So this graph is just depicting our length distributions. So for this project, we chose to capture fish within 10 millimeter length bins. And so this is over all years, 2010 to 2020, and has all three species. The one I definitely want to point out is largemouth bass, being that they are the larger of the three. You can really see the length distributions as it goes across the entire graph. That doesn't mean that the other two don't have good distribution, because they do. Largemouth bass, you can just see it a little bit better. All right, this graph is depicting the distribution of ages for all species. Again, bear in mind that this is for all years collected. Again, I'm just going to point out largemouth bass. If you'll notice that there's a lot of outliers for largemouth. That is probably due to 2020 data, as we collected some older fish that were in that 12 and 13 range, and then we were able to collect smaller fish. And so they're filling up that gap of 1s and 2s and 3s. So the next couple graphs I'm going to show you are the frequency of ages, and just kind of so you get an idea of how they're distributed. So as you can see, for largemouth bass, we now have a good distribution. We have ages all the way to this 1.1 age, all the way down to 13. Bluegill follow that same pattern where we get the younger ages, and then we get a couple individuals all the way up into 6 and 7. And then for Black Crappie, they also now follow the same pattern where we get these 1, 2, and 3 ages, and then we're also being able to fill in gaps of 7 and 8. So having all these ages, we'll be able to see exactly what factors are affecting fish growth when we start analyzing and running our covariates with increment measurements. Okay, so this next section, I'm going to be specifically focusing on my biochronology or annual growth section. Bear in mind, this is all preliminary data. We are still working on getting things entered in a usable fashion. So this is just some stuff that we have at this point. So look at the biochronology, we're using linear mixed effect models, and we're looking at how factors are influencing increment growth. So here, increment is meaning otolith annuli measurements. So that's going to be those measurements I took using mica, where you go from like the nucleus to the first annuli to second, and so on and so forth. And then our fixed effects are going to include age, which is age of increment. So wherever that increment you randomly choose is, say the increment is in the middle and it's 5. Again, that's just an example. And then we also have ACC, which is age at capture. So that's going to be what you age the fish out to be at the end of the otolith. We also are including some random effects, including individual fish and year. We're also looking at cohort. Cohort is going to be birth year. So that is all fish born in a certain year, whether that's 2010 or whichever year. Here are some graphs kind of depicting whether growth was good one year or over another. First, I'm going to point out the 0 or the dashed line. So anything above this dashed line is going to be a good growth year. Anything below it is going to be a poor growth year. Also, everything that is circled in green ovals is good growth for that species, as opposed to red ovals, which are poor growth. You'll notice that all 3 species have different years of good growth, and for largemouth bass and black crappie, they definitely have poor growth in 2019. You can see that bluegill did have poor growth, but it wasn't quite as distinctive as black crappie or largemouth bass. So moving forward, we will be adding different covariants to our linear mixed effect models, including vegetation abundance, zooplankton abundance, water level, and potential water quality data. These will be added as our fixed effects. So the next couple of slides are just kind of an overview of the other sections that I will be working on. At this point, we don't have analysis done of year class strength or mortality and individual growth, but I will be working on them in this coming year. So for year class strength, we'll be using age to determine which years were good growth years and which were poor. and then looking at what factors were going on. And we can also compare this back with our linear models as well. And then I will also be looking into mortality and individual growth. I'll be utilizing catch data from the Emiquon monitoring program at IRBS, creating age length keys and plotting catch curves to determine when mortality is happening. Moving forward, I will be collecting samples both this month and next. We are hoping to capture the 2020 drawdown and see what kind of effects that's having on our sport fish, positive or negative. It'll be interesting to see. I'll be continuing to collect covariant data. I will be starting the analysis with the covariant data and adding that all in, as well as starting the analysis on my other sections of this project. And with 2021 field collections, that means there will be more otoliths. So I will be processing 2021 otoliths as well. At this point, I'd just like to thank everybody that's helped me with this project, including Illinois River biological staff, both current and present technicians, the Nature Conservancy, and University of Illinois Springfield. Questions?